and also the antibodies that interact with it to block the virus's infection. I thought the summary that Ed gave was extremely good. Okay, so this is the hemagglutinin from the Hong Kong influenza series that began in 1968. And it simply shows that the molecule has two functions. It binds salic acid, as you've heard, and it also mediates membrane fusion between the virus membrane and the endosomal membrane. These sorts of structures are obtained by removing the membrane anchor from down here at the base of the spine to get a soluble protein for crystallization. And the receptor binding site is colored here in blue. It's, I'll show you in a moment, it consists of a conserved pocket of amino acids surrounded by variable residues. And the membrane fusion is part of the function of this red part of the molecule here. And it's primarily involves this, well, it involves a whole lot, but it primarily involves directly this fusion peptide at this position here. Okay, so you'll see the molecule's a trimer, and this particular subunit is multicolored to show you where individual domains are delineated mainly on an evolutionary basis. And the red part is mainly to do with membrane fusion, the blue for receptor binding. Okay, so each of the subunits, which are identical, are made up in the hemagglutinin of infectious particles of two polypeptides, HA1 and HA2, and they're made as a precursor, which is cleaved to generate the carboxy terminus of HA1, which is located here, and the amino terminus of HA2, which is the amino terminus of the fusion peptide, which is located there. Okay. There are 16 subtypes of hemagglutinin, which are defined antigenically, structurally, and from the point of view of sequence. And you can see on here that the 16 are split into two groups, group 1 and group 2, and group 1 contains 3 clades, group 2 contains 2. Okay, so that's the 16. All of the hemagglutinins look very similar. They've all been solved now. They're all in the structure, all in the database. These are 5 representatives of the 5 clades. And you can see they're very similar. There are a number of structural features that you can see even on a slide like this, which delineate the groups. So for example, this small alpha helix here, which together with the long alpha helix form the main part of HA2. This small alpha helix in H3 and H7 HAs, which are members of group 2, are shorter than the small alpha helices in group 1 HAs. So here for H1, and here for H9, and here for H13. And also when the two helices are joined together by an extended loop, noted here, and in the case of group 2 HAs, this linkage is a shallow linkage into the terminus of the long alpha helix, whereas in the, and you can see that there in the case of H7, and in the case of group 1 hemagglutinins, it's a taller turn, which is at the amino terminus of this guy. Okay, and that's the case for all of the group 1 hemagglutinins. And then if you look at the top of the slide, this is the so-called 190 helix, which I'll mention again later. You can see that in the group 1 hemagglutinins, this helix has more or less the same orientation at the top there, whereas in group 2, the H3 and H7 examples here, it looks like it's twisted, and in fact the whole domain has turned through a variable number of degrees depending on the strain, but peculiar to the members of the 2 groups. Okay. So the other glycoprotein of the membrane, as you heard from Ed, is the neuraminidase. And I think the neuraminidase, which varies just about as much as the hemagglutinin during during a pandemic period, has a justifiable claim to be a component of any vaccine. I mean, many of the vaccines that are produced now, certainly in particular viruses, have very little neuraminidase representation. But you can see that neuraminases also are divided into subtypes. There are 9 neuraminidase subtypes, and they again form 2 groups, with the 1, 4, 5, and 8 forming group 1, and the others forming group 2. They're also defined into these positions of subtypes and groups on the basis of structural differences. And that's pretty simply shown. The neuraminidase is a tetramer. This is viewed down, looking onto the virus surface. And the neuraminidase top here, like a mushroom top, is joined to the virus membrane, as you'll remember from Ed's slide, by a stalk. So there's a stalk projecting from the undersurface of this into the virus membrane. Each of the subunits has an active site for neuraminidase action, and the main structural difference between members of the 2 groups are in this position here, this loop called the 150 loop, and denoted by that residue at 149 and 151. All of the members of group 1 have this open loop, whereas the members of group 2, this loop is closed like so. So in this particular piece of the slide, this is N1 again, showing an open loop. And in yellow and green here also is R2 from group 2, N2 and N9. It turns out that during neuraminidase action, this loop, even in group 1, snaps shut to assume the same structure as this, but this is used as a structural distinction between members of the 2 groups. Okay, so enough of that. The 2 functions of HA, the salic acid binding and membrane fusion, I'll look at in the next couple of slides. The salic acid binding pocket, as I said, is this blue part of the monomer that's colored there. And this is a magnification with this short alpha helix, which I pointed out on a previous slide, shown here in line form, the 190 helix. And the secondary structure elements that form the site are this 130 loop, which is this line here. In some cases, the 150 loop, which is this line here, but not in all cases, so it's not marked here. The 190 helix and the 220 loop. So they're basically the conserved secondary structure parts. And the yellow part here is salic acid itself. Salic acid is bound in all of these hemagglutinins in basically the same way. And the interactions that are formed are with semi-conserved residues. For example, this in the 130 loop, the carboxylate of the salic acid, which sticks into the site, forms a hydrogen bond with a hydroxylated amino acid at position 136, and sometimes a hydroxylated amino acid at 137 also. And with the carbonyl, the peptide carbonyl of residue 135, there's an interaction with the nitrogen of the acetamidose substituent of the salic acid, which lies over another conserved residue which forms the base of the site, this tryptophan 153, and their hydrophobic contact between the methyl group of the acetamidose substituent with this tryptophan. The other conserved residues that form the base are this histidine at 183 and this tyrosine at 95 in this case, which is brought up by the folding from the bottom here. And these 3 are hydrogen bonded together to form a sort of bit of a network. There are other conserved residues, in particular this chap here at tyrosine 194 and 195 rather. Okay. you'll hear later that one of the main distinctions between avian influenza viruses, where all of the subtypes are found, and human influenza viruses, where we've only experienced viruses of 3 subtypes since viruses were known. They differ as far as receptor binding goes on the basis of recognizing salic acid in different linkage to the galactose, which is the penultimate sugar in receptors. In the case of the human viruses, the linkage is a 2,6 linkage to the galactose. So you could just to orient you on this for subsequent things, that's carbon 2 there. And so that would link to the exocyclic 6 carbon in the case of the human, so-called human receptor, and it would link to the 3 carbon of galactose in the case of the avian receptor. So I'm sure you'll be shown that again by both Robert and Joshua, but I'm not going to say anything more about it at this stage. Okay, and the second function then is the membrane fusion activity, and this is just another way of orienting a hemagglutinin subunit with this time these colors are color coding for a conformational change, which is triggered at the low pH in endosomes, usually around 5 to 5.5, although it can go up above 6 depending on the particular strain. And I think you can follow and probably have seen elsewhere where these individually colored components finish up. Basically, the fusion peptide at pH 5 is extruded to form a structure whose main objective seems to be to put the fusion peptide and the membrane anchor region together at one end of a newly formed structure, which is largely a rod. The HA1 part, the receptor binding region of the hemagglutinin, is dissociated into 3s. The extent to which it's dissociated is eventually not totally clear. It's probably not as dissociated as this. But the structure of that component can be obtained by simply cleaving this off in the low pH form. And it's the super determined by crystallography is basically superimposed with what it is in the neutral pH structure. Okay, so I don't think I need to mention too much about the conformational change. Basically, it involves extrusion of the fusion peptide up to this sort of location, and that's followed by the short alpha helix, which turns through 100 degrees, and importantly, this extended loop which links the 2 helices in the neutral pH form becomes an alpha helix at low pH, and that is sat, the whole new helix is sat on the top of this gold part here, or yellow, I guess it is, this yellow part, which is basically unchanged between the neutral pH form and the fusion pH form. And then below that, at residue 106, it turns out and it's shown by these 2 green turns of the helix, they refold into this turn, and that's how the rest of the molecule finishes up running position the membrane anchor at the same end as the fusion peptide. Okay, so antigenicity. Well, when you heard about the international surveillance network, which the WHO has for tracking influenza viruses, or influenza as a disease, and over 100 labs around the world are involved in isolating viruses from outbreaks of disease and characterizing them. They can characterize themselves, send them to the international centers, of which there are 5. And when they get to the centers, at least, and often when they're in the national labs, they get their hemagglutinins at a minimum get sequenced. And the sequences indicate, if you look from in this case from the start of the H3 pandemic in 1968, for let's say 10 years, the number of substitutions in the hemagglutinin shown here in black largely, are located on the three-dimensional structure. Okay, and if you look at the subset of those which are passed on to subsequent to the hemagglutinins of viruses isolated in subsequent years, you find that they seem as if they're focused around the receptor binding site. The ones that were down here towards the fusion active part, the fusion active part have basically dropped out. They're not fixed. And the positions of these amino acid substitutions more or less correspond to, with a much smaller number, with the positions of amino acid substitutions identified in monoclonal antibody selected mutants. Okay. So the points where those amino acid, the positions where those amino acid substitutions are located on the structure are the sites, indicate the sites at which antibodies bind. And that's shown for these three monoclonal antibodies. And there's no reason to imagine that it isn't the case for all the antibodies that are produced when you get immunized. You'll see that these two in particular bind over the receptor binding site. And this one binds about 15 angstroms below the receptor binding site. They all block receptor binding, and they all seem to neutralize virus infectivity by doing that. And these two do it much more efficiently than this one, about 3 logs, between 2 and 3 logs better. This one doesn't block very efficiently as an FAB. It blocks as a full IG. You can imagine the full IG is this sort of size. The FAB itself doesn't block very, very efficiently. Okay, so the idea that sterically in some way, this one, even though it binds outside of the site, blocks receptor binding, it seems okay. These seem to block directly. The antibodies like this penetrate deeply into the site sometimes. And a number of these have been identified by people, by a few groups actually, to not only, and the feature about those particular antibodies is that they seem to have a fairly long HCDR3. And to some extent, the sequence of that CDR in some cases also looks as if it's mimicking salic acid. But anyway, that's an idea that has been had. But the significance of those penetrating antibodies is that they seem to be subtype specific. That is, they're not strain specific like these guys are that were isolated from the 1968 hemagglutinin. These that are penetrating deeply into the receptor binding site are sometimes subtype specific. Subtype specific is, I think, from the point of view of cross-reactive antibodies, which we'll consider in a minute, subtype specific antibodies are pretty important. I mean, the problem that Ed spoke about in terms of predicting what next year's virus is going to be would be solved if you didn't have to predict that. If you had if you could induce antibodies which interacted with all of a subtype. Now they wouldn't interact with the next subtype, which is the next pandemic, but then you can't predict that anyway in any reasonable way, since it comes likely from an avian source through another animal or something like that. Okay, so these have attracted quite a lot of attention, being cross-reactive within a subtype. There's another one which has been observed, which is also cross-reactive within a subtype and also binds quite tightly, but it binds on the back of the receptor binding sites. It binds sort of here. And it interacts with this one is an H5 specific antibody, and it interacts with all members of the H5 subtype that have been isolated from humans. Okay. So the whole business of looking for cross-reactive antibodies that would be able to protect against any influenza virus has had quite a lot of steam, and a lot of antibodies have been isolated, which are cross-reactive to a degree. I'm going to talk about two from isolated from the Corti-Lanzavecchia group in Switzerland, which have turned out to be the most cross-reactive, particularly the second one that I'll mention. So this was the first one, and the hemagglutinin is labeled essentially as it is along, and you see that this antibody binds close to the short alpha helix in the red subdomain of the hemagglutinin that's involved in membrane fusion. That position in vitro, it can block the cleavage of the precursor to HA1 and HA2 that I told you about, and it can also block the conformational change required for fusion. So in vitro, those activities are potential mechanisms of action of these antibodies in vivo. In that case, they would block the membrane fusion of the virus on the way into the cell if they stay as stable complexes into the endosome, and they would interact with hemagglutinins displayed on the cell surface late in infection, so they block infection at the beginning of infection and at the end of infection. The idea at the end of infection, also when the hemagglutinin is displayed on the cell surface, is that the antibody hemagglutinin complex can also be recognized by Fc receptors. Fc receptors on cells which are able to lyse infected cells. And it may well be that these activities through Fc receptors are the major way in which this particular group of cross-reactive antibodies that are recognizing the membrane fusion region of the hemagglutinin, it may well be that that's the way that they mediate virus neutralization. Okay. The antibody which is more cross-reactive than any other that's been studied is compared here, this MEDI8852, because it was compared in a collaboration with MedImmune and was owned by MedImmune, is compared here with the one from the previous slide, the FI6. And the FI6 was really more group 1 specific than totally cross-reactive. It reacted with a number of group 2 hemagglutinins, but not with all of them. In the case of the 8852, the antibody reacted with all 16 of the hemagglutinins. And part of the reason for that might be, is shown on this slide, where the bound antibodies to H5 from group 1 and H7 from group 2 hemagglutinins are superimposed. And they basically superimpose extremely well. They're in the cleaved parts, they're only 2 and 1.5 angstroms distinct. By comparison, in this antibody that was less effective as a cross-reactive antibody, these equivalent regions are 11 and 13.5 angstroms apart. So the degree of similar interactions is much greater in this case than it is here. Okay, and in addition, all 5 of the CDRs, the heavy and light chain, are utilized by the one that's completely cross-reactive. And if you compare where it touches the hemagglutinin, shown in orange and green here, with where group 1 specific and group 2 specific antibodies bind, and you'll see that this one that cross-reacts with all basically combines these 2 areas, and this blue square and this red square. So basically, it seems to bind as a sum of the 2 antibodies that are known from others' work to be group specific. Okay, and then the last slide that I want to show you is obtained, in this case, by cryo-electron microscopy. And cryo is going to be used and is already being used to analyze antibody complexes with hemagglutinin, and also to define, by studying individual HAs from drifted viruses, what, if any, differences in structure can account for the differences in antigenicity. And one of the things about the membrane-anchored hemagglutinin, which is the membrane anchors here in a detergent micelle, one of the things about the linkage to the hemagglutinin is that it's a flexible linkage. And we could describe that pretty effectively by looking at an antibody complex, yet another cross-reactive antibody, which binds in this location. You can see it's a slightly different orientation from the previous antibodies that I showed. And that antibody had the distinct advantage operationally for us, in that instead of these guys varying around this flexible linkage, it sort of kept it in this position. And you can see in purple the flexible linkage between the ectodomain of hemagglutinin and the membrane anchor. Turns out that this antibody also blocks infection, but only as an FAB. And I guess the idea then might be that it's only the FAB that can penetrate down to this position between spikes on a virus particle. If that's the case, it may well be that these cross-reactive antibodies block activities on the virus rather than requiring FC receptors to function in neutralization. But I would still go for FC receptors. Okay, so basically I'll stop there. I think it's worth noting that one of the things that cryo-electron microscopy gives you clearly are the carbohydrate side chains on the molecule. And that's different sometimes from the crystallographic data, because the carbohydrate side chains are removed in order to get crystals. And also it's sometimes possible, like at the moment with the hemagglutinins, it's difficult to crystallize them anyway. And so the ability to analyze these things directly by cryo-EM is going to be important, and has already given the same sort of resolution, less than 3 angstroms, that you obtain from X-ray crystallography. And the people who are involved in this in Mill Hill over the last few years are listed here. Thank you.